Yes, Mike.
Okay, good afternoon to everyone. We're This afternoon we're supposed to witness the defense of matter examination defense of Mr. Reinier Xander Ramos with his thesis entitled "Dynamics of a Nonlinear Cellular Automata Model of Biological Neurons and Applications to Young and Aged Neurons," with Dr. Johnrob Bantang as advisor, Dr. Banzon as reader, Dr. Esguerra as examiner, and Dr. Gennevieve as examiner. Okay. So we'll have 45 minutes for the presentation. We should be relatively uninterrupted except for clarificatory questions, and be followed by an open forum after 45 minutes. Okay, let me begin.
So, good afternoon, everyone. Again, I'm Reinier Xander Ramos from the Complexity Science Group, Instrumentation Physics Lab, under the advisorship of Dr. Johnrob. And today, we will learn about the dynamics and stability of a nonlinear cellular automata model for biological neurons with applications to young and aged neurons. Sorry. So shown here are different degrees or levels of complexity within the nervous system. The brain is perhaps the most complex organ in the nervous system and even in the universe. And all throughout the years, the greatest challenge for neuroscientists is to understand how this brain works. How can it analyze and memorize new information? on all the levels, not just in the brain or in the organ level, but as well as in the tissue level and even in the neuronal level. This thesis presents an alternative approach of investigating or understanding these neuronal dynamics. Specifically, I want to answer the following questions. How does a biological neuron fire? How do we build a computational model for this neuronal firing? How do we extend the model to a neuronal network, especially in large-scale networks? Is this model computationally fast and efficient? How sensitive the model will be to various initial conditions and parameters? And how do we utilize the model for application in experimental or biological data? In the early 19th century, Camillo Golgi developed a black staining method in order to deduce the morphology of the central nervous system. Using his method, Ramon Cajal was able to prove the existence of these tiny indivisible units, which we now refer to as the neuron. Each neuron consists of the following parts. The shorter branches are called dendrites as the receiver of an input signal. And by the way, this input signal is usually in the form of an electric signal. The electric signal will then travel through the soma or the nucleus of the neuron, where the signal is interpreted either to be relayed or not to neighboring neurons. If the signal is to be relayed to the neighboring neurons, then the signal will travel to these longer branches called axons, right? Usually these are myelinated to aid for a faster transmission of the signal. And at the end of the axons are these spaces or junctions called the synapse. Here, the electric signal is converted into chemical signal. And then the chemical signal, which is in the form of ions, are converted back to electrical signal to the neighboring dendrites of the neighboring neurons. In this synapse happens the spike or starts the spike generation. So usually when you probe the brain using your EEG or electroencephalogram, you will see this reading or this recording with a series of spikes after the initial stimulus. Looking closely with the singular spike, we see this is the voltage versus time for a single spike inside the neuron. In particular, the neuron maintains a quiescent state or a resting potential of around negative 70 millivolts, and we refer to this as the quiescent state. Okay. In addition, inherent to the neuron is a threshold voltage, usually slightly above the resting potential. Okay, and when the input signal does not exceed this threshold voltage, then we will have failed initiations, meaning no spikes are generated. But if the input signal exceeds the threshold voltage, then the neuron will transition from quiescent state to the firing state via depolarization. Okay? And usually this firing state is a positive voltage difference of around 40 millivolts, and we call that the action potential. So here we have the resting potential, and here we have the action potential. After that, the neuron will repolarize and transition from the firing state to the refractory state. And in the refractory state, the voltage reading is usually below the resting potential, around negative 120 millivolts. Okay. And then, after that, the neuron will transition from refractory state to the quiescent state via hyperpolarization. Okay. Specifically, this hyperpolarization, this polarization process happens across the neuronal membrane. Okay. So the generation of spike anatomically is mediated by these voltage-gated ion channels across the neuronal membrane. And two particular channels are very influential to the spike generation, the sodium channel and the potassium channels. And some smaller chloride ions happens to leak out or in to the neuronal membrane, influencing again the voltage difference across this neuronal membrane. And so this difference in ion concentration can also be modeled via an electrical circuit shown in this diagram, okay, where we have the conductance for the sodium channel, the potassium channel, and the leak channel. Okay. This electrical circuit is utilized by Alan Hodgkin and Andrew Huxley in 1952 as they developed this set of 4 ordinary differential equations. So by the way, these are coupled ODEs. Okay? So here, they were able to map the voltage over time as a function of the input current or an external stimulus. M, N, and H are just the activation and inactivation probabilities for the sodium and potassium channels. Okay. They were also able to experimentally determine these constant variables for this set of equations. And the alphas and betas in the activation probabilities are governed by the Boltzmann transport equations as shown here. If we numerically solve this four set of ODEs for a single neuron and for varying input currents, we will obtain this membrane potential or the voltage over time plots shown on the left. So here, the topmost represents the voltage over time for a very small external stimulus that is 2.5, and here 2.5 microamperes per square centimeter. And then we increase the external stimulus and we see variations in the spikes generated. Okay. We can count the number of spikes given a period, and that will be our firing rate R. Okay. If we plot our firing rate R as a function of input current, we will obtain this profile we call the activation function of the neuron. So again, this is the numerical solution for a single neuron using Hodgkin-Huxley ODEs. Experimentally, Prescott et al. Was able to derive as well the same profile of the activation function, but for various excitability classes. So they were able to discover three classes, the first of which is class 1, atonic spiking. Here, the firing rate and the input current has a continuous relationship, meaning if we just increase the input current, we just result to more spikes generated. So no phase transition is observed. However, in class 2 or in phasic spiking neurons, there is a discontinuity at a minimum input current. So here, spikes is generated only at after this minimum input current. So there's a one-phase transition from a no spikes to a series of spikes after this minimum input current threshold. And finally, there's a few of the neurons experimentally that they found are class 3 single spiking. So here, the spikes are generated only at very, very high input currents or external stimulus, shown here. Okay. The Hodgkin-Huxley ODEs is just one of the many models of single neuron spiking, and it was developed in 1952. The motivation of their ODEs is derived from the original integrated fire developed by Louis Lapic in 1907. And in this integrated fire, we just have one ODEs, and it's just your typical electromag equation for capacitance. Okay. However, in this ODE, Lapic needed to manually reset the neuron from the firing to the recovery or the refractory state. So there's a manually setting, or there's a influence by the user or by the experimenter for the integrated fire model. However, that resetting is already incorporated in Hodgkin-Huxley. That's why the HH model is more powerful than the IAF. Other models, the latter models, Fitz-Hogg-Nagumo and Morris-Lekar models, are just derivatives of HH wherein they want to simplify the 4 ODEs into a fewer number of differential equations and for specific applications. But what's most common for all of these single neuron model spiking is the profile or the properties of the neuronal activation function. So common to all of them is are the thresholds, the minimum input needed, A sub 0, and a maximum input, A sub 1. And in between this A0 and A1 is a monotonically nonlinear relationship between the firing rate and the input current. There's also a maximum output, A sub 2. And we can model the nonlinearity using a parameter B. Okay. So since this is the usually the most common properties for the neuronal activation function, then we can generalize it and normalize into this nonlinear approximation as shown in this figure. Okay. So again, we incorporate all of these thresholds, A0, A1, A2, and B, on our equation. And for our purposes, since we have 4 parameters, we want to reduce the number of parameters, then we just set A1 equals to 1 for all the results later on. Okay. Now again, this is a activation function. This is an activation function for a single neuron. What if we want to build now a network of neurons? So one way is to arrange these neurons in a lattice, as such, shown here in the left, right? And then we connect each neuron according to these directed edges shown here. Okay. And then, what if we try to apply or solve the the Hodgkin-Huxley ODEs for each neuron? So now, we have 4 ODEs for each of these neurons. So we will have to solve 36 ODEs for this 9 neuronal network. And then plot the voltage over time here on the right. So it's promising, right? It's possible to simulate a neuronal network using the HHODEs. However, when we want to upscale our network, say 1,000,000 neurons, we have to solve 4 million ODEs that are coupled with each other just to investigate the dynamics of that neuronal network. And that takes time and even greater computational resource. Throughout the years, so specifically that HH network was constructed by Pang et al. In 2014 using these coupled input currents on the neuronal network. Okay. But other models for neuronal networks have also been established. One is the Wilson-Cohen model, which uses 2 ODEs coupling the excitatory and inhibitory neurons. And one is the Kuramoto model, which uses coupled oscillators on their model. And again, what we see is the common profile for their activation function, similar to the single neuron models. Okay. Since solving a large-scale network of HHODs is very tedious and needs a higher computational resource, we want to propose another approach using cellular automata. Specifically in this thesis, we just use a square lattice with a lattice size L by L. Okay? And each of the cells that forms within the lattice is analogous to a neuron. Okay? So we will have a population of N equals L by L. And then we assign each of the neuron a state. The states can be quiescent, firing, and refractory. And if you remember, these are the spiking states when you generate a spike during neuronal firing. Okay. How about the synaptic connections? So we apply more neighborhood for each of the neurons. So say, for example, this cell on R will have a more number or 8 more neighbors around it defined by the shaded red. How about for a cell on the edge? So it will only have 3 neighbors. We can apply lattice boundary condition, for example, a toroidal periodic boundary condition, such that we connect all the cells on the leftmost column to the rightmost column, and we connect the topmost row to the bottommost row. In this way, the cell on the edge of the lattice, the cell F, will have its regular 8 more neighbors defined by the shaded blue. And finally, we applied the interaction rules. So these are the rules that dictates how each neuron behaves. And we map it using the spiking regimes that were shown or that was discussed earlier. So if the neuron is in the quiescent state, it will only transition into firing state if there are exactly two number of firing neighbors in its neighborhood. Every firing state neuron will transition to a refractory state, and every refractory state will transition into a quiescent state. Okay. So for example, we initialize the lattice with the set of neurons at t equals 0. So if we apply this set of rules, say for example this Q, since it has exactly 2 firing neighbors, then at t equals 1, or the next time step, this will transition into an F. As opposed to this Q here, this has exactly 3 firing neighbors in its more neighborhood, so this will remain quiescent, or remain in Q state. All the F's will become R, and all the R will become Q. And so, at t equals 1, this is now the configuration of our lattice. If we apply this numerously or as necessary, as t goes approaches infinity, then we will obtain this following spatiotemporal activity. So here we have the lattice size L equals 100. Okay. So in general, we can state the algorithm for constructing a CA as follows. So we start by constructing and populating the lattice by n equals L by L. So this is just for square lattice. Okay. At t equals 0, we randomly assign each cell with a state from the set S. Okay. And for each cell, we update the state via the interaction rules governed by the set R. And again, this set R is influenced by the neighborhood and lattice boundary conditions defined by the sets L and N. Okay. And then we repeat these steps as necessary until a steady state is observed. So in this case, in a Brian's brain case or the Brian's brain CA, the steady state are gliders. So the set of rules that we have seen before, so this set of rules are called the Brian's brain rules developed by Brian Silverman in 1996. Okay. So it's just a derivative of the original CA, the first CA, the Conway's Game of Life. Okay. This Brian's brain CA is a discrete neuronal CA because we are just using discrete states, Q, F, and R. In reality, neurons can be in a continuum of states because the voltage values can be of any value. Okay? So, we want to propose this nonlinear neuronal CA with a continuous state. Okay? So, we consider a population of n, again, equals with a lattice size 100 by 100. And each of the neuron in the cell, in the lattice, can have a state A, which we define to be the net activity of a single neuron. And this A can take a value, a uniform random value from 0 to 1. So that at t equals 0, we can generate a uniform random sampling of this A as shown here. Okay. And for the interaction rules, we set that the input to each of the neuron to be the average activity A of its neighbors. So if it has more neighbors, then you will take the average A of those 8 neighbors. That will be your A sub in. Okay. Then we will use the nonlinear approximation that we have discussed earlier and map it to get or to obtain the A sub out. So the A sub out will be the next state of that neuron at the next time step. However, we can vary this neighborhood. So we can apply various neighborhood conditions. So previously, we have seen this outer totalistic Moore. We can also consider a totalistic Moore setting where the cell itself is part of the neighborhood or part of the average. Right? And we can also consider a fraction of the population to be injected by external stimulus, besides the input current from its neighbors. We can also vary the lattice boundary conditions. So we have seen earlier the toroidal, but in this thesis, we also considered a spherical lattice via Mercator projection. So we just fitted a square lattice on the sphere surface using this projection. And we also considered multi-layered toroidal NCA from 2-layer to 4 layers. And we classified the resulting steady state behavior as follows. So here, shown here is the neuronal patch average over time for the whole lattice. Okay, so the patch average for the whole lattice over time. And shown here is the average steady state for the class 0 or quiescent steady state. Meaning, as time goes to infinity, the average activity becomes 0. Okay? And we have 2 types, a fast decaying to that 0 steady state and a slow decaying to that 0 steady state. For each of the plots, we compare the steady state activity behavior using totalistic and outer totalistic, and we see that this setting does not affect the classification. Okay. And we also observe a spiking steady state, which we refer to as class 1. Okay. And here, instead of going to 0 as time approaches infinity, the steady state activity goes to a non-zero number with two types, a low activation and an activation. Shown here are the spatiotemporal activity for each of the subclasses. So for class 0, or the quiescent steady state, we have this fast decay to 0 or to inactivity, and a slow decay to a 0 spiking or 0 steady state. And here we have the the spatiotemporal activity for the spiking steady state with low activation and high activation. So the colors here represent the neuronal activity for each neuron. Now we compare with the different or the various neighborhood and boundary conditions that we use. So here we compare the toroidal versus spherical boundary conditions, and we see that our classification is unchanged. Okay. So shown here is a spatiotemporal activity, a sample activity from class 0B with toroidal and spherical lattice boundary conditions. Next, we compare for each n-layered NCA. So again, we see that the steady state behavior is unchanged even if we increase the number of layers from 2 to 4 layers. However, when we consider or when we introduce an external stimulus to the lattice, the class 0 behaves or transitions from class 0 from 0 steady state to a spiking steady state. And we need, it seems to be that at least 5% is needed to be injected with a constant input current so that the activity is sustained over the lattice. So shown here is active spatiotemporal activity for class 0B, where 1% is injected, so we see that the activity still decays. But when we increase the fraction of neurons injected with external stimulus to 5%, then we see that the activity is now sustained, even in 10%, shown here. So now we can play around by varying the morphology or the topology of our lattice. But how do we do, or how do we compare with the original HH network? So shown here is the simulation time versus the population size that we consider. Okay. The blue lines or the blue triangles is the simulation time for the using ODE solvers for varying population size. And we observe that the simulation time using forward Euler method increases quadratically as we increase the population or the number of neurons. Using much accurate ODE solvers like RK4 or Rijikuta fourth order and the Livermore algorithm or LSODA just increases the simulation time, but the profile is not changed. However, using our model, the cellular automata model, the simulation time increases linearly with the population size. So this allows us to simulate a large-scale neuronal networks that is much faster and efficient than using ODE solver for the ODEs. Now we look at the sensitivity or the stability of our system by varying the initial conditions and the parameters. Okay. Specifically, we analyze the local stability and global stability of the system of our activation function. The fixed points are defined to be the steady state solutions of the systems, and usually they lie on the diagonal A out equals A in, or X equals Y. So these fixed points or the steady state solutions of the system always lies in the diagonal. Specifically, we can obtain the fixed points by the intersection of our function to that diagonal. So here are the fixed points for this specific activation function. Now, if we vary our initial state or our initial condition, over time, it should stabilize to one of these fixed points. We could analyze those using the cobweb diagrams. So say for example, we initialize at this state, right? We draw a vertical line from that point to the function, and then we draw our horizontal line from that point to the diagonal. We repeat these steps until we reach a fixed point or until some oscillatory or limit cycle is observed. In this case, we reach these fixed points, and so we say that this fixed point is a stable fixed point. We do this for all of our classes or classifications from class 0A to class 1B. And we analyze as follows. So in class 0A, the activation function is usually completely below the diagonal. And we just have one fixed point. And it is located at 0 or at the origin. And so any initial state that we choose will stabilize at 0. Hence, a collection of these neurons will likely result to a quiescent steady state. And even, it will be a fast decay because any initial state will just transition towards 0. However, in class 0B, we have 3 fixed points. Okay? And initial states below the second fixed point takes shorter paths stabilizing at the first fixed point, as shown by these 2 cobweb diagrams. But initial states above the second fixed point stabilizes at the third fixed points and takes longer path. Hence, a collection of these neurons will most likely transition to the 0 quiescent state or the quiescent steady state. Why? Because it will only take these initial states to move towards 0 at a very shorter time than those states on the upper half of the activation function. Okay. So the initial states here are overwhelmed by the faster trajectory of these 0 quiescent steady states in this region. Hence, this will result to a slow decay, right, in the slow decaying to the quiescent steady state. In class 1A, we have only 2 fixed points. And the second fixed point is usually located usually located at a low value less than 0.5. Any initial state that we choose except at 0 will stabilize at that second fixed point. And hence, we have a low activation spiking steady state. In class 1B, we also have 2 fixed points. And the second fixed point is usually at a high value, greater than 0.5. So any initial state that we choose will stabilize at that second fixed point, resulting to a high activation spiking steady state. So here's the summary of the fixed points of the systems. Okay, so for class 0A, we have 1 stable fixed point, stabilizes at there. In class 0B, we have 2 stable fixed points and 1 unstable fixed points, meaning the system, if the initial state of the system is just along this neighborhood, then that system will move either towards the third fixed point or the first fixed point. And for both class 1A and class 1B, we have 1 stable fixed point and 1 stable fixed point. Now we consider analyzing the global stability of the system. For this, we will use 2 diagrams, phase-space diagrams and bifurcation diagrams. In the phase-space diagrams, we plot 2 parameters with each other. So here we plot B versus A2. For a fixed A0, so we fix the last parameter. And then we color each pixel by the average steady state for the whole lattice. So now we're just not considering single neurons, but we consider the stability of the whole lattice. Okay. So black pixels or pixels that are closer to black indicates quiescent steady state or class 0, while colored pixels that are closer to the yellow one indicates spiking steady state or class 1. And then we do this for varying parameters. And it's shown here. So the topmost row, we compare A2 versus A0. In the middle row, we compare B, the nonlinearity B, and the input threshold A0. And in the last row, we compare B versus A2. And moving to the right, we increase the other parameter. Okay. Let's look at it individually. So in the topmost row, we can see or we can observe that the active phase region, these colored pixels, increases as nonlinearity B increases. In the middle row, the active phase region increases as the maximum output A2 increases. However, in the bottom row, the active phase region only increases when the minimum input A0 decreases. We can summarize the phase space diagrams as follows. The activation functions with threshold parameters covering more of the region above the diagonal or between the diagonal and the activation function will most likely result to a spiking steady state or class 1. Okay? So as we increase the nonlinearity B, this activation function will plateau more on A2, right? If we increase the maximum A2, maximum output A2, then this region also increases. And if we moved A0 to the left, if we decreased, we also increase this region, resulting that this region will most likely result to a spiking steady state. Now, in the bifurcation diagrams, we plot the steady states again of the whole lattice as t approaches infinity for varying parameter, where we now fix two of the parameters. Okay. So shown here is the steady states for varying B, where we fix A0 and A2. If you obtain all the possible bifurcation diagrams, we will obtain these diagrams. So from left to right, we increase the A2. From top to bottom row, we increase A0. And again, each plot, we increase B. The black dots represents the simulation data or the average steady state of the lattice, while the solid gray line indicates the unstable branch that we observe from the cobweb diagrams. However, this unstable branch is not observed in our bifurcation diagram. So we just these plots by this unstable branch. And this means that the unstable branch of fixed points are overwhelmed by the average of the whole lattice when we take into account the whole lattice as one. Okay. Additionally, the average steady state increases with nonlinearity B. And this is again due to when we increase the B or the nonlinearity parameter, again, the activation function will plateau more on A2. And this results to a faster transition from low to a higher output activity. Now we apply our model to some biological or experimental data. Shown here is the firing rate versus injected current for young and aged neurons obtained from the slices of prefrontal cortex of rhesus monkeys. Okay. And with this, we can observe that it has the same profile that we have seen earlier, the activation function, right? So we can fit, we fit these, their data points into our nonlinear activation function as shown here. So the parameters are given. And then we use this as the interaction rules for our nonlinear neuronal CA. With that, we obtain this neuronal patch average over time shown in this figure. The darker lines show the original young and aged neurons activity, so the darker red and the black, while the lighter colors, the pink and the gray, are neuronal CA injected with external stimulus of 5%. What we found out is that younger neurons need 5% of the stimulus, again, similar to what we have observed earlier, to sustain the activity, shown here. While aged neurons responds with 4 times the stimulus. So we only have 5% stimulus, but the response of the aged neurons is greatly amplified 4 times. But that's for the continuous NCA. We can still use our discrete NCA by considering that the activity A, to be the probability of the neuron to fire, okay? So that we can utilize the discrete states QFR from our spiking state regimes. And with this, we obtain the following discrete neural response for the young and aged neuronal systems. Okay? So these are the discrete state over time for young and aged, which are now comparable to the recordings that we can obtain using EEGs. The animations below are the spatiotemporal activity of this discrete neural response for the whole lattice. Okay. So we observe that high spiking activity indicated by the F states here, the yellow ones, are prominent to aged neuronal systems. And again, this for younger systems, we need at least 5% of the stimulus to be injected in order to sustain some activity in this younger neuronal system. To summarize, a biological neuron undergoes stages of spiking states when firing. These states represent different potential difference or voltage difference that the neuron exhibits. The HH model describes this potential difference via a set of 4 ODEs, and every single neuron model that we have seen displays similar profiles of the activation function in which we normalize into our nonlinear approximation. And then we utilize a cellular automata model to build large-scale networks of neurons with the activation function as a rule, resulting to the following steady-state behaviors comparable with the HH model. So we have a class 0 quiescent steady state for the nonlinear neuronal CA with fast decay and slow decay. And we also have a low and high activation for the spiking steady state or class 1. Our model, the CA model, is faster than solving ODEs regardless of the scale or the population size. And our model can even simulate 3 orders of magnitude more neurons higher than the Blue Gene P supercomputer, which uses utilizes the HHODEs originally. And our model also requires less machine memory as compared to the Blue Gene supercomputer. Locally, each neuron follows the cobweb trajectory and stabilizes at the fixed points of the activation function. But globally, these unstable branches of fixed points are not observed when the average of the network is accounted for. So they're being overwhelmed by their neighbors. The experimental firing rate from the young and aged neuronal systems is fitted into our activation function, and the dynamics of this NCA with this activation function is analyzed and is comparable with experiments as shown by the discrete neural response. So that's it for my research work. Thank you for listening, and I hope you learn about the dynamics and stability of nonlinear cellular automata model for biological neurons with applications to young and aged neurons.
Thank you, Reinier. Congrats for a very good presentation. My question is actually more of a request to clarify slide slide 58. Was it 56? Slide 56, please. And compare it with 58 or slide 6. How can you discuss why you proposed that the QFR to Q, back to Q, is the correct sequence of the states? And maybe you can discuss it together with the activation function, just to make it clear. Bakit Q to F, F to R to Q?
Oh, okay.
Yeah.
Thank you for noticing, Dr. Bantang. So I use this specific transition or this series of transition because it is derived from the spike generation, right? The spike regimes. So here again, we normally the neuron has or sustains a quiescent state, right, or quiescent potential, and then if the input signal to the neuron exceeds the threshold voltage, then the neuron will transition to F. So that's why we have that Q to F. And then all firing neurons will immediately repolarize to the refractory period or the refractory state, hence the transition F to R. And then this F to R, during the refractory period, the neuron will start to hyperpolarize of their ion channels, right? And it will go back to transition from R to Q. So this is that series Q to F, F to R, and back to R to Q. Okay.
Slide 49.
Okay.
Yung second bullet, you use the phrase will most likely.
Yes, sir. Okay.
Why do you have that hedge phrase? Will most likely. Ano yung exceptions?
Oh, okay. Thank you for the question, Dr. Esguerra. We use most likely because we believe that a more exhaustive search for varying parameters is needed, right? Because we only vary the parameters A0, A2, and B from 0 to 1 with a step of 0.1 for each. But some systems can have islands of stability or instability, even if we increase the resolution of the parameter space, right? So this doesn't take into account those islands of stability or instability. That's because we have not yet observed that. So maybe in the future, we can increase the resolution of our parameter space for our exhaustive search in order to confirm or to conclude a more solid conclusion with this.
So is this nonlinear cellular automata supposed to replace HH eventually?
Okay. I would like to say no, because what we want to do here is just propose an alternative approach. It does not have to replace the HHODEs or the other models, right? Because here we still don't know yet what really is A or the parameter A for our model, right? We just normalize everything from 0 to 1. So this A, the activity can be interpreted as the probability to fire. It can also be interpreted as the voltage, exactly the voltage. But again, that differs from model to model. So we just generalize them into one, but it doesn't have to replace all of those models. Right. Yeah, shown here. So here we have the activation function for each of the models. So specifically for Hodgkin-Huxley, they just calculated spikes per second for varying input current. But here we have frequency over input current. And then for Maurice Lecar, we have again frequency. So this frequency is somewhat similar to the firing rate, but it's not exactly the same. They're not exactly the same. Okay? As well as this spike frequency for the Fitz-Hugh-Nagamon. Yes, sir. Even these models does not aim to replace the Hodgkin-Huxley or even IAF. They just these are established to be able to simplify the HH model with specific applications. For example, the Maurice-Lecar model is usually applied to the neurocortex or the neurocortical neurons, the neurons on the surface of the brain, while the Fitz-Hugh-Nagamon models the activity of pituitary gland and hypothalamus. So they have different applications as well.
Okay, let me modify the question. Is there a measure of so maybe it's not supposed to replace it. So is it supposed to emulate the Hodgkin-Huxley model? Maybe it's not supposed to replace it. Is it supposed to emulate? And in that case, how do you measure what's the fitness of the emulator with this particular choice of A0, A1, A2, whatever? This is the best choice of how do you assess that the fitness as an emulator?
That also depends on the system that we are modeling. Because
Now, suppose
yes,
you already know the system
Yes.
that you're modeling. So in that sense, how do you say these are the best choices for your fit parameters?
Okay. So I think I can. That will be here. 58. Nope. Okay. So I think I can give an example for that, specifically when we applied it to young versus aged neurons. So again, the firing rate that was obtained uh, experimentally has a similar profile. Okay. However, uh, in this region, if we have an injected current beyond 300, I think that's 350 picoamperes, right? We still don't know what happens to the firing rate of the neuron. So we are limited by this window, and so we can normalize it to fit to our activation function. But that doesn't mean that the parameter thresholds that we obtain are the best ones for it, because again, that is limited by the data that is being fitted to our activation function. See? Because different experimental data have different values or continuity of values for the current as well as for the firing rate. Okay. Some neurons, for example, in the squid giant axon from the original Hodgkin-Huxley, they have microamperes, in the order of microamperes. But in the rhesus monkey, they have in the order of picoamperes. So it's varies differently from each species or depending on the biological sample. Yes. Yes, and we can compare it in reality using the proposed dynamics or the observed dynamics in the steady state and even in the spatiotemporal activity. So this is what we can compare with uh, biological data.
Thank you for the
Okay.
presentation, and
Thank you.
congratulations to you and Dr. Bantang for the publication of the results. So my first question is on slide 55, the results for the nonlinear. Yeah, 55.
I 1.5.
Oh, sorry,
Okay.
50, yeah, yes, 55.
Okay.
55.
55.
Yeah, 55.
Okay.
And I'd like to compare that with the one in slide 32 because um, the spike, no, the trend is going up and then
Yes, slowing down.
it's
Going.
stable.
Okay.
But for this case, you have a spike for the age neurons with external, and then for the young neurons with external, there's a deep compared to the one in slide 32. So could you explain what's why the trend is very different?
Oh, compared to
To
slide
slide 32.
33.
32.
32. Okay.
Yeah.
Mhm.
For the 5% injected class 0 B
For the
case.
5%.
Yeah, 5%.
Yes. So, okay. So I think for here, the activation function, actually, we can see in the previous slide that the activation function for the younger neurons is really almost is completely below the diagonal and very far from that diagonal. So if we try to do cobweb diagram from that, so for example, we say we initialize at here, it will immediately go to 0. So every initial state from here, we immediately go to 0. So a collection of those neurons um, will be what do you call this? Will steady state or will transition to a zero quiescent steady state almost immediately, right? Because the activation function is very, very far, but is completely below the diagonal. Okay, and when we inject external stimulus to 5%, we see here that the average activity for the whole lattice is just that 5%, meaning all of the neurons without external stimulus, their activity still goes to 0, but the average activity that is sustained is just from the external stimulus. So that's why we have this here. On the other hand, I think slide. Okay. On the other hand, in here, we also started with a uniform random distribution, right? And specifically for these specific parameters, the activation function is much closer to the diagonal as compared to that. So where can I show that example? Wait lang. So this specific parameter, so that's 0.1, 0.7, and 2. So if we compare here, so that will be 0.1, 0.7, and then a nonlinearity 2. So that activation function is much closer to the diagonal. And so we can observe that some of the initial states will stay longer when we draw the coupled diagrams, will stay longer in the spiking steady state before transitioning to zero. That's why it takes longer for them to steady state into the zero quiescent steady state. Unlike this type of activation function, that's very, very far from the diagonal. Mhm. Hmm. I think with regards to the the lattice formulation or the network formulation, the neighbors will can affect the neighboring neurons, but there's an inherent synchronicity within the network. So if most of your neighboring neurons transition to the quiescent steady state, you are most likely to transition into quiescent steady state as well, because of due to synchronicity from the neighboring neurons. Yes, that happens for the young. So if your neighbors have very low activity, close to zero, when you take the average of your neighbors, it's also close to zero. So that's why you synchronize with them, right? As opposed to if your neighboring neurons have a non-zero spiking steady state, so if you take the average, most likely you will have also a non-zero state and the next time step. Okay.
In connection to that, may I know if you also calculated different percent external stimuli for this case?
Yes, po.
So what is the trend for the other, for greater than 5% and lower than 5% external stimuli for the young neuron and aged neuron case?
Oh, for the young versus aged, we haven't yet explored. So maybe we can look further into it. So we just considered 5% actually. Actually 1% and 5%. And we found that at least 5% is needed um, for the steady state activity of the younger neurons to become non-zero. So we just ended up there. So I think, yeah, that's a good point that we should explore on various uh, fraction of external stimulus here. Okay. Not, no, sir. Di po. Not in this young and aged system, sir.
I was wondering what are you comparing in terms of time complexity in slide 63,
Ay,
where you have your model against the ODEs.
I think that's 45. Okay.
63 ata yun.
Ay, ito po.
Ay,
Ano na? Ito po.
61 pala. Yeah.
Okay.
Now, you were comparing your model against what was one where you use
The Blue Gene supercomputer. Okay. So actually, it's in this slide, I think. There. So here, we are comparing the machine specs of that is used in our work as compared to the Blue Gene supercomputer. So the machine used for this thesis has the CPU and GPU and this RAM, 16 gigabytes. And since we were able to simulate up to 10 to the 6 neurons in just one CPU, so that's our uh, neurons per CPU. And for memory or for memory usage, we just divide this population for the RAM that is used, so we obtain the 16 kilobyte per neuron. However, for Blue Gene supercomputer, which utilizes the HHODEs actually, they build a neuronal network with these HHODEs. They use this almost 147,000 CPUs and 144 terabytes of main memory. So if we calculate for CPU, they can simulate just 6,000 neurons per CPU. So that's very, very few as compared to the 10 to the 6 neurons per CPU in our model. And with respect to the RAM usage, so uh, it follows that they have approximately 144 kilobytes usage per neuron as compared to our 16 kilobytes per neuron. So this comparison makes our model more efficient, or shows that our model is more efficient than using the HHODE network.
About a comparison of separate, you're comparing two separate quantities, right? You're comparing your model, which is different from what's being solved by the gene.
Uh, yes po. Mm. 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 Yes. Mm. Inefficient. Yes. The CA model. Yeah. Yes po. But in this plot, the simulation time versus the population size, uh, the ODE solver or each of these line of data points here is simulated using these specs. So the same specs. Okay. And, and so yes. Opo. Mm. Opo. Yes. Opo. So, yes po. So, added to that, for the same machine, we were able to simulate up to 10 to the 6 neurons using our model, but only up to 10 to the 4th number of neurons using ODE solvers. So, anything beyond this, actually the machine or the computer hangs up and then shuts down. So we are really unable to evaluate the time here because it just shuts down and restarts. So, and overheats. Okay. Yeah. Mm-mm. Yeah, different machines. Okay. Yeah, I think. Okay. Thank you for the question. Yung dynamics po mismo. Actually, okay. Wait. I think it's in here. Okay. So, okay. So this, I think I can answer that with this slide. So here is the voltage over time calculated in the neuronal network of 9 HH neurons, right? Using various ODE solvers, the RK4, the SODA, and forward Euler. So here we almost see almost no difference using ODE solvers, but again, this has a definite value for the voltage over time. Okay, we can compare it to the discrete neural response that we have obtained, right? So, for example, in here, so that is comparable to the discrete neural response that we obtain from our system. Right? So again, this already adds up stochasticity because we treat A as the probability to fire, right? Yes po, this is just for one neuron. So that activity is comparable to one neuron on the plot uh, on the ODE base here. Okay. So same shape. The most important thing here is that they have the same spiking profile, right? The spiking shape. Yes, sir. I think this periodicity of this arises from the exact solution of the HHODs because they already, if numerically solve it, it's exact. But again, in reality, that periodicity is not exact, so we can expect that there's, for example, in between these spikes, there might be more of quiescent time before spiking again. Mhm. Yes. Ah, um, yes. This. So is an example of, yeah, an experimental spike generation. So uh, like here, right? Mhm. Yes po. Uh, more closer, yeah. Actually, the discrete neural response that we have obtained is much closer to this series of spikes than that of the exact solution of the periodicity from the ODEs, actually. Sa collection, it's in a patch. Yeah. Mm. Yeah. It's really harder. It's really difficult to probe just a single neuron. So maybe it's a small patch of maybe 9 to 10 neurons or just a sheet of neuronal tissue. That's,
Yep. Do we have further questions?
I may question po from the audience.
Yeah.
more on clarification lang. Sabi kasi sa motivation kaninang study is generalize yung model, tama ba?
Oh, okay.
Ayun, is generalize yung model, pero from what you said earlier, kasi is naging alternate way lang siya ng current model. So, can you clarify kung paano siya naging generalize? I guess.
Okay. So, actually, it's just an alternative approach. I think I misspoke about the generalization. What we meant by that is that all of the activation profiles, right? There. So all of the activation profiles here can be quote unquote generalized by normalizing, right? Normalizing the x-axis and the y-axis from 0 to 1. And with that, we can obtain the normalized approximation or the nonlinear approximation of our activation function. Okay. So this now takes into account or can be compared to each of the models separately, but not as a whole. So that's what we meant by that. Okay. So because again, the parameter A can be interpreted as a voltage or the probability to fire. So it's like a Reynolds number, right? Where you can, it's, what do you call this? You can know the laminarity or the size of the tube or everything about fluid dynamics in the Reynolds number. So this is somewhat like that number. It's just a unitless value. Okay. Ay, sir, si Doc Macam po.
I have a question about slide 17.
17. Okay.
So the model of the spike, you have the QF and R states.
Yes,
So I'm
sir.
wondering if you can simulate the dip in the R state, because in your model you have the probabilities for Q to F state and F to R and then R to Q.
Yes,
So I'm
ma'am.
kind of expecting the dip state somewhat like the HH network from Q state to R state. Sorry, F state to R state.
F state to R state. Oh, okay. Actually, this here in the HH network, this is already a result of the firing rate over time for many neurons, right? But that transition from F to R is cannot be observed in this activation function because that's just on the actual voltage versus time. Okay. The transition, I think it's here. Okay, so these transition states from Q to F, F to R is just obtainable from the voltage over time or the neural response. So this is different from the neuronal activation function. So there are two different things: the neural response, the actual neural response, and the activation function or the response function. So there are different things. Did that clear up your question, Doctor? Okay.
Okay, thank you.
Thank you.
So,
Ranger, so yung so yung neurons mo, nilagay mo sa nodes ng nilagay mo sa lattice, no?
Yes, sir.
Paano kung ring lang? Paano kung nasa isang ring lang yung arrangement? Meron ka bang ina-anticipate na mangyayari? Di ba parang kasi alam mo, di ba, kung ano mangyayari at the level of a single neuron.
Mm-hmm.
Ngayon, let's say nasa ring lang kasi sa tingin ko itong lattice mo ngayon, medyo komplikado nasasabi mo lang. After
Yes,
mo ginawa, ito nakita ko. Kaya kung
Okay.
nasa ring, ano yung ma-anticipate mo na behavior?
Actually, with that ring, there will be a fewer number of neighbors for each neurons. So if we consider a toroidal neighborhood or toroidal moor, then we'll just have two neighbors, right? The left and the right. So with that, maybe what I think is that yung specific parameters for the activation function like is 27. Okay. So if we consider these specific parameter thresholds, this will now result to a maybe result to a different steady state. But I think we can still classify each of those set of parameters in one of these classifications. So it can still be a quiescent state or spiking steady state. However, this may be changed into a spiking steady state or so. Right? Yes, for the total activity. Um, hmm. I think what I see is that if we consider a ring, then we will form similar to the gliders in the Brian's brain. So the specific set, depending on the initial state of the configuration, if it stabilizes to a set of various configurations of this A or neuronal activity A, right? So you will see, I think we will see similar to the gliders in the Brian's brain. Yes, in one dimension. Yeah, it's like similar to a wave or like a pulse signal that's just moving around the lattice or around the ring. So yeah, but I think it's interesting to look at maybe. Actually, closer to that, I think, is this spherical lattice, because in the spherical lattice, the way we generate the boundary conditions, again, we fit a square lattice on the sphere surface, meaning on the poles or near the poles of that projection, all of the neurons are connected, right, on the topmost and the bottommost row. So if one of the neurons is firing, then that firing activity or that activity will just be transmitted to all of its neighbors in that along that ring. So we can isolate this actually as a ring uh, and can be compared to that. In biological sense, the sheets in the neurocortex is somewhat like this actually. Okay? So because in the outermost sheet or layer of the brain, almost all of the neurons are what do you call this, fully connected. But they're still connected to the neurons below. That's why we have this spherical lattice. But I think it's the closest one that I can think to your question.
Questions? No?
Okay.
Simulation of the more neighborhood, because you simulated more neighborhood, and then you have the outer and inner case.
Outer
So
totalistic.
what's the physical? Sorry, yeah, outer totalistic and totalistic more. Yeah.
Okay.
What is the physical significance of the totalistic more?
Oh, okay.
Yeah.
Thank you for the question. I think in biological sense, this totalistic Moore setting is closer to the real neurons because inherent to each neuron is a threshold voltage, right? So um, uh, in that sense, the neuron itself should be part of the neighborhood or part of the interaction rules, right? So I think the totalistic Moore setting is more applicable or more closer to the real neurons than the outer totalistic Moore. In this work, we just compared if we tried this outer totalistic versus totalistic setting, we just analyze if there's any difference. But it turns out there's not much difference in the resulting steady state.
Except for the fast
Yes. So in here, there's just a slower, or what do you call this? Faster decay for the outer totalistic when we consider the outer totalistic setting. But again, this is what do you call this? Uh, this has minimal effects on the overall spatiotemporal average. Because if you consider a large network, especially in the human brain, I think this dip here will vanish, right? Because here we just consider 10,000 neurons, right? So if we consider a larger number of neurons, specifically in the multi-layer, then that okay. So this is for different layers. That dip will vanish.
But if it will accumulate, wouldn't it accumulate instead? Or
Um,
how will it cancel out?
hmm.
If you have many small, you have a small difference, and then you have more
More
neurons,
of
and
the
then all
small
of them will add
difference.
up, and it will become more obvious in a larger scale.
Hmm. I think. Oh. Yeah, you have a point there. Yeah, when you accumulate small differences, then that should decay faster, right? What's the name? Okay. Um. Right. Okay. Mhm. Yung 100 by 100 lattice. Toroidal lattice. Mhm. Yes. 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 Actually, some neurons does really have these self loops, especially mirroring neurons, okay, or gate neurons. And with that, it seems like um, they have this self feedback, right, on their self, where their output becomes a part of their input as well. Okay, so um, yeah, I think, but just some of just a specific type of neurons in the brain. Okay. But overall, I think even if we increase the number of neurons, I think there will be less the difference in the steady state between outer totalistic and totalistic will be. I think it's I think this there will be a minimal change, or yeah. But I want to get back. I want to find out actually. I really want to find out. So that's really a good. Yes. So I will have to simulate again for that to account for that question. Thank you. Uh, uh no, not much difference. So, well, wala. Hindi naman. I think what's more important there is if we try to add weight for the cell itself. if its weight contribution to the neighbors is different from the neighbors. Because so because if you just add itself as part of the neighbors and they have equal weights, then if it has low activity, then supposed to be it has it follows that it has also lower contribution to the average, as opposed if it has higher activity at that time step. So I think we can consider weighted average when we count for totalistic mode. I think that's more applicable. Yeah, to test the significance. Yeah.
Yeah. So
Fine. Thank you,
I
ma'am.
guess we have exhausted the questions. Okay. So will we have a break? Yes. Okay, so we'll have a break. So
Okay.
thank you
Thank you
very
po.
much.
Thank you everyone for the question.